Dear students, I welcome you all back to the new video of cell cycle. I hope that you remember that we have covered 2 to 3 topics of cell cycle, like the chromosomes, their different types. Then we just touched the topic of cell cycle, and then we did the process of mitosis, which is one of the types of cell division, in detail in our last video. We had a gap, so now I'm here with you to discuss another type of cell division. It is termed as meiosis. This is a type of very lengthy sort of cell division. So I have divided the whole topic into 2 videos in which we will be covering meiosis, significance of meiosis, and the meiotic errors. So let's start the video with meiosis. So, what is meiosis? Meiosis is basically a type of cell division in which the parent cell, which is undergoing the cell division process, is basically producing the 4 daughter cells, which are all haploid in number. Now, you must be thinking, where is this happening? So, this type of cell division is happening in our gametes, the sex cells or the germ cells, which are termed as sperms in males, while eggs or ova in females. This is also happening in the spores, like small mother cells. They are producing the spores, which are also haploid in nature, and they undergo the same process of meiosis, in which the parent cell is dividing into 4 daughter cells, which are haploid in number. Here, the question is, what is meant by haploid? So, if you look at your structure, then you're having 46 chromosomes in every cells. But the cells, the germ cells that are forming as a result of meiosis, they're haploid. 46 means the diploid number of chromosome. If you reduce it to half, you will get 23. So all the daughter cells that are producing as a result of meiosis division, they are haploid number, which means in case of a sperm cell or the egg cell, you're having 23 chromosomes, which is haploid number of chromosome. I hope that this is clear to you. Now I will be discussing in detail different events of meiosis. First phase of meiosis is basically meiosis I, which is also termed as first meiotic division. This is also called the reduction division. Then we are having meiosis II, which is the second meiotic division, and it is same as that of mitosis. So it is also termed as mitotic division. Well, for your ease to understand the whole topic, I have just added few important terminologies which you will see like probably most of them in the first phase of meiosis I, which is called the prophase I. So with the explanation, I will cover all of them, like tetrad, homologous chromosomes, bivalent, synapses, crossing over, chiasmata, everything we will discuss. I will also show how is crossing over is happening. So let's start meiosis I. In case of meiosis I, karyokinesis I, like the first nuclear division, what is happening, what phases are there. So we are having prophase I, metaphase I, anaphase I, and telophase I. Sorry, I just forgot to mention one here. So this is telophase I, remember. If you look at the phases of meiosis, then we are having multiple sort of stages or subphases in case of prophase I, which is the longest phase of the whole meiosis division. So we are having different sort of substages like leptin, zygotine, pegitine, diplotin, and dikinesis. I'll be explaining you with the help of this illustration. So here, first we'll discuss the first subphase of prophase I, which is termed as the leptin. You must be remembering that the cell is basically an interface first, where it is just assembling every material for the cell division to start. So once the cell enters meiosis I, prophase I, then some drastic changes are happening. How? Before starting this division, you must be remembering few things. So here I will try to tell you about homologous chromosomes. Now, homologous chromosomes are what? Homologous chromosomes are those chromosomes which are having the same gene sequence throughout their chromatids. Okay, so we are having the homologous pair here. One pair, you are just getting of the pairs you are getting from your father and the other from your mother. So homologous chromosomes are those chromosomes which are like similar in their shape and sizes and in their gene sequence. So in case of leptin, what's happening? The chromatin material is just condensing and it is just forming the homologous chromosomes, which are thread-like structures, and they are just beaded. Okay, so they form leptin at this stage. At the leptin stage, the leptins are formed, which are the beaded chromosomes, the beaded homologous chromosomes. So this is the first stage, leptin of prophase 1. Then these homologous chromosomes, when they pair up together, then this is basically termed as synapses. Okay, the homologous chromosomes, they pair up together, they come closer to each other and they form the synaptonemal complex. So they are just pairing up here. This is termed as synapses. Okay, so here you can see the lengthwise pairing of these homologous chromosomes. Here you must be remembering that in this synapses, what is happening? You are just having one bivalent here and the other bivalent here. If you just count the number of chromatids, then you are having 1, 2, 3, and 4 chromatids. So this is basically the tetrad. Okay, and individually they are bivalent to each other. In case of peg-18, what is happening? These homologous chromosomes are overlapping at each other and they are forming a sort of linking point together, which is called the chiasma or chiasmata somewhere. So in peg-18, they are just like making the chiasma. Now in case of diplotin, these bivalents, they are just trying to separate from each other. Still they are joined together at chiasma, and at this point they will do the crossing over. Now what is crossing over? Here you can see these are the two homologous chromosomes. One of the homologous pair is having this same sequence of genes ABC here on the first chromatid, and the other sister chromatid is also having the same number of chromosomes, the same sequence of genes. Okay, sorry, same sequence of genes on it. And this is another homologous chromosome, and it has also like having the same sort of sequence of genes. You can see here the small alphabets ABC. So they are homologous chromosomes and they have just paired up together. Then here you can see they are overlapping each other at some point. So this point where they are overlapping each other, this is termed as chiasma or chiasmata. Now after just crossing over, when they separate in dikinesis stage, what happens? They are just recombinant chromatids of the homologous chromosomes, and this is the non-recombinant chromatid. So in case of diplotin stage, these bivalents, they are attached at the chiasma. They are not completely separated. But at the end of prophase 1, what happens at the stage of dikinesis, these homologous chromosomes, after crossing over, they separate from each other, and this process is termed as terminalization. You can see here easily that these bivalents are attached here, but in dikinesis they have separated, right? So this results in crossing over, and these homologous chromosomes, they are now no more same. They have shown some recombination at this point. Okay, now they are just totally separate from each other, and this nuclear envelope also disappears. Nucleus, that also disappears, and the spindle fibers are formed just because centrioles have moved to the opposite poles. Now what will happen? After prophase 1, metaphase 1 will start. In this, the tetrads will line up at the equator or the center at the metaphase plate. Okay, so they are linked. You can see easily that they are linked with the spindle fibers. After metaphase 1, what will happen? The anaphase 1, which is another important stage, it will start. Now this homologous pair, they like this is basically separating from each other, and this is the point where the reduction division is starting. You can see here that the number of chromosomes, they are reducing to half. After anaphase 1, the telophase 1 will start, and at the telophase 1, these homologous chromosomes, they have reached their poles, and the nuclear membrane reappears, nuclei basically reappear, and the centriole is formed back again. You can see the cleavage here because the cleavage is taking place, and at cytokinesis 1, this cytoplasm will also divide and the two daughter cells will be formed, but they will be haploid in number. You can see the reduction here easily. So this is the end of meiosis 1. After that, there will be short resting phase, which is called the interkinesis, that will be separating both meiosis 1 and meiosis 2 from each other. But this is not very lengthy, sort of long span period. This is very short period, and after a short period, meiosis 2 will start. So we will start meiosis 2 in the next video. This is it about meiosis 1. You just went through the whole process of meiosis 1, and meiosis 1 is, if you look at the stages, then it is basically divided. The karyokinesis 1 is divided into 4 phases: prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 1 is a lengthy phase, and it is subdivided into 5 different substages. One is basically the leptin stage, in which leptins are formed, which are the beta structure. Then we just saw the zygotine, in which the pairing up synapses is happening, and of the homologous chromosome, that is basically happening. Then we had the pegitine, in which there is chiasmata formation happening, and in diploidine stage, the crossing over was taking place, and in dikinesis, the separation of homologous chromosome, that is termed as a terminalization, because at this stage the two bivalents, they just separate from each other in the form of a zipper. Like if you want to open the stand of the zip, then as you just open it, so they just they are just separated from each other. So it's the same way those bivalent will be separated from each other, and this process is termed as a terminalization. So after dikinesis, we are just our cell just goes to another sort of phase, which is called the metaphase 1, then telophase, then anaphase 1 and telophase 1, and after that the cytokinesis happens. In the next video, I will tell you all about meiosis 2. So till then, you take care. Bye.